All right, guys, this is technically the third part of Chapter 15's notes. I know that we did part 2 kind of in person. So I wanted to go ahead and do at least part 3 here. And in part 3, we're going to get into a little bit of the behavior of genes that are located on sex chromosomes, as well as what happens when you either alter chromosome number or chromosome structure, which are all kind of fascinating things when you get down to how they work. Now, as we go through some of this stuff, I'm going to gloss over some of it, and come back and expound on it later, provide a little bit more detail on some of the topics that might be maybe a little bit more interesting or high interest. Okay, so let's go ahead and get started here. The section 3 here deals with sex linkage, which we've talked about a little bit already. They do exhibit unique patterns of inheritance, and so those are some of the things that we've kind of gotten into and talked about with pedigrees, that a lot of the inheritance patterns that you see with sex-linked inheritance can be snuffed out in pedigrees because the inheritance patterns are very unique. Okay, so we've also at some point at least talked about sex determination. Okay, and for human beings, determination of sex, meaning whether or not it has male parts or female parts, happens because of the XX and XY system. Okay, and so we don't necessarily need to revisit it, but I want you to recall and be reminded of the fact that human males have an X chromosome and a Y chromosome, and human females have 2 X chromosomes. Okay. Other organisms. Now, I don't think this is anything that you need to worry yourself about. It's not going to be on my test, and I don't think it's going to be put on the College Board test. But other organisms have different unique ways of determining the sex of their species. Okay, so you see there with this little insect, this grasshopper guy. Get my mouse working. There it is. The grasshopper guy. They have what we would call the X0 system, where the females are determined the exact same way that humans are. 2 X's equals a female, and a male is just the lack of another chromosome. So if they just have an X chromosome, that means they're male. That's how that works for them. The little bird here, little rooster. And I don't think this is specific for chickens. I think this is more for birds in general. Use what's called the ZW system. And it's almost like backwards. It's almost flip-flop from what ours is. And you notice that the female has a Z chromosome and a W chromosome. Okay? And so that would be akin to our males having an X and a Y, whereas the males of the bird species have 2 Z chromosomes, which would be akin to our females. Okay? I think the most interesting of the 3 that we see right up here is the bees. Bees actually have a very unique system where their males are haploid and the females are diploid, meaning that all of the cells in a male are haploid and all of the cells in females are diploid. It's something it would be hard for us to wrap our mind around. Okay, it's effectively every kind of the way you might look at it is every cell in the male bee's body is sperm, kind of. You know, that you can imagine. It's be gross and weird, but that's how it would work. Anyway, I spent way too much time talking about that, but I thought it was really interesting, and the fact that you've got all these different types of creatures that determine their sex different from ours. Okay. We've talked about sex linkage at least a little bit, right? The fact that most of the genes of importance are located on the X chromosome. And the reason for that is because the Y chromosome found in the males is really, really, for the most part, insignificant in terms of having traits on it that mean or anything or matter. It does have a relatively important gene on it called the SRY gene, which stands for the sex-determining region of the Y chromosome. And really all that gene does is code for testes in the male. It gives the male parts, as we might say. There are other genes on the Y chromosome, but they're relatively insignificant and definitely don't need to be talked about here. Okay, so that has implications on how traits on the X chromosome are passed on to male children. Okay, because the male children's not going to have a second copy of that chromosome to override the other one that mom would have given him. Okay. So genes located on either sex chromosomes are called sex-linked. And yes, there are some that are located on the Y that are other than the SRY, but they are relatively insignificant. Okay. So in addition to some genes being inherited on the X chromosome, you do see quite a few disorders and diseases that are inherited on the X chromosome. Oftentimes these are recessive in nature, and I've listed a couple of them here, and we've talked about them. They've been in examples that we've done already. So we don't need to spend a lot of time on them here. If you have questions about them, I'd feel free to ask them. I might not know the answer, but I'd be happy to try and find it. Okay. There is a really cool phenomenon in the females called X chromosome inactivation. Okay, so we're talking about the behavior of sex chromosomes. And in mammalian females, there is a phenomenon known as X inactivation, in which one of the two chromosomes that the female have will randomly inactivate during embryonic development. So if you go to every cell in the the mammalian female during their development, their embryonic development, you will find that one of the X chromosomes randomly just shuts down. It inactivates, and it creates something called a bar body. And the bar body basically is this shriveled up little X chromosome that doesn't do anything. Okay? And so, for the most part, this doesn't play a big role in females that might be homozygous for a trait, but it definitely plays a role in females who are heterozygous for particular traits, because if you are looking at a specific trait that, let's say, codes for the color of fur, and that's the example we're going to see in the next one, the color of fur, one of those chromosomes, let's say one chromosome has the allele for black fur and the other chromosome has the allele for orange fur. One of those chromosomes will actually just shrivel up and effectively die and not ever be expressed. Its allele will never have any bearing on what the animal looks like. So we see this a lot in calico cats, and I don't know a lot about cats. I'm not really a cat person. I know I prefer dogs. Some people will hate me for that, but whatever. But I prefer dogs. But what happens in calico cats, the way I understand it, is you will almost never find a cat, a calico cat, that is male. Because calico cats have this fur color, like we kind of see right here a little bit. That's a kind of an example. Have this fur color because of the fact that they are females. Okay? They have this patchy black, patchy orange. Sometimes there's some white in there as well. Fur color because of this X chromosome inactivation and because of the fact that that cat happened to be heterozygous for the fur color. Okay, so during early embryonic development, this is what the chromosomes would look like in the cat as it starts to develop. They'd both be active. Okay, you'd have an orange one and a black one, but as cell division starts to occur, and the embryo starts to develop more, different patches of cells will have had one of the chromosomes inactivate. Okay, it will deactivate. For instance, this chromosome here, this little balled up kind of chromosome that has the black allele. That chromosome is inactive and it won't ever be expressed, which means the only chromosome left in that particular area is going to be the orange one. And so every cell created from this one, so as the organism divides, it gets more cells, those cells divide. So as this cell divides, the only chromosome that's left on is going to be the orange one in every succeeding daughter cell as well. So you get these little splotches of orange fur because those derived from cells in which the black chromosome had been deactivated. Whereas in this area, like right over here, at some point early on in development, the orange chromosome had been deactivated, and the only one that was left to be expressed was the black one. And so as this cell continued to divide, the only one that was left on was the black one. So every little cell in this little region would have had black fur because the black chromosome was the only one that was still on. We learned, and you know, I learned something new every year. I learned last year that it is in fact possible to get male calico cats, and I believe the reason for this is because of non-disjunction, which we will talk about in the next section. Okay, so that might be something we can revisit as we work our way through. Now, this is also seen in human females. Okay, and I'll show you a picture when we get back together in person. But human females actually have a situation where some of their cells in their skin will deactivate the sweat glands. And it's a really weird type of mosaic feature where different splotches of human female skin will literally sweat while others will not. And you can kind of map it based on tracking sweat beads and stuff like that. So I'll show you a diagram of that. It's quite fascinating, and the reason for that is because different chromosomes that coded for sweat glands in a certain part of the body deactivated, while others in other parts of the body stayed on during early embryonic development. Kind of a neat phenomenon. Okay, so that brings us into Section 4. Section 4 discusses the alterations of chromosome number and structure. Okay, so the chromosome number and structure can be altered in many, many ways. We can have breakage of chromosomes. We can have reordering of chromosomes. We can have whole-scale chromosome, large-scale chromosome alterations. We can have cells or embryos that have too many chromosomes. So there's all different number of things that can happen to the chromosomes in developing zygotes and embryos. Okay, one of the first things you need to understand is that large-scale chromosomal alterations oftentimes lead to miscarriages. Miscarriages actually happen far more often than you would ever think, oftentimes without the mother ever even knowing. And so that's a way, nature's way of saying there's too much wrong with this baby as it's developing. We have to start over, or else the baby will be born, you know, with some debilitating type of things going on. So miscarriages are actually happen quite often. Okay, they are less common as the baby gets older and older and older, which is why when you hear about a miscarriage of somebody who had a late-term miscarriage, their baby might have passed away when it was almost fully developed, which is obviously very shocking and saddening. Whereas other people might not ever know that they had a miscarriage because it happens maybe so early on in embryonic development. Okay. Oftentimes, alteration of chromosome number is due to a phenomenon known as non-disjunction. Okay. And non-disjunction is where pairs of homologous chromosomes don't separate normally during meiosis. So this goes all the way back to chapter 13. And we have non-disjunction events that can occur in a couple of different places, and depending on where they occur, I say places, I mean phases of meiosis, and depending on where they occur can lead to much more severe cases of non-disjunction and what we call aneuploidy. Okay? So a non-disjunction event could occur between meiosis 1 and meiosis 2, in which the homologous chromosomes themselves do not separate, as we see here in this diagram. The homologous chromosomes never separated during anaphase, and what that leads to is a much more serious case of non-disjunction than the other side over here. Because what we see when we finally get down here to the gametes is we see that this sperm or egg might have one extra chromosome, this sperm or egg might have another extra chromosome than it should, and these two here are missing a chromosome. So in this case, every gamete that was produced in this event is screwed up. Okay? Whereas this other scenario, the non-disjunction event happens in meiosis II, where the sister chromatids don't separate. And that's distinctly different from the top instance, which I talked about. And this leads to less wide-scale what's the word I'm looking for? Screw-ups. I'm just going to go with that. Screw-ups. Less wide-scale screw-ups. Where we see 2 of these, sperm or egg, are still normal. That's what they should be. Whereas just these 2 over here, one's got an extra and one's got less. So these non-disjunction events mean that something has not separated the way it normally should. Okay? Also, we see breakages of chromosomes that can lead to 4 types of changes in chromosome structure. We see deletions, duplications, inversions, translocations. These are things I'll touch on a little bit more in detail as we go through. But I wanted to first touch on, what did I do here? Messed that up. Yeah, right here. This diagram here shows you what happens in these breakages of the chromosome material. A deletion will basically just remove a giant chunk of DNA, and along with it, anything that would have been, important in the development of an organism. So if there are 3 really important genes here in this chromosome segment D, now they're all gone. And that baby will not have those genes to develop and won't express that trait. And sometimes that's innocuous and nothing happens to the baby. Other times it's so bad that the baby has to be spontaneously aborted in the womb. By the body. Duplication takes certain segments and just repeats them over and over again, and sometimes that can be just as devastating as deletions. Inversions is where you take a chunk of the chromosome, you pull it out, and you flip it around, and you stick it back in there. Okay, and assuming that no transcription machinery or transcription code is messed with in any way, that won't always be as debilitating, but oftentimes it can be, because sometimes you're reading things in reverse now, and maybe you get a defunct or messed up version of the protein that should have been in there. Okay, and then we have translocations, where translocations are kind of like crossing over between 2 totally different chromosomes. Okay, so like here we see this might be called chromosome 1, and maybe this one here is called chromosome 2, and we actually get a physical switching of DNA just like crossing over, only they're not homologous. They don't share the same gene segments, and so that can cause issues as well, cause disease as well. Alterations of chromosome number result in what are called polyploidy or aneuploidy. Okay, polyploidy manifests as multiple sets of chromosomes in organisms, and this happens a lot more often in plants than it does animals. I'm not going to spend a lot of time on it, but this little animal right here, this little rat guy, he's actually got, how we say our chromosome or our cells have 2N, right, which is our normal set number. We have 1 set from mom and 1 set from dad, and they're found in our cells, right? This guy's got 4N. Okay, somehow he's got twice the normal amount of chromosomes that he should, and he was able to live through it, or his species was able to live through it. That happens far less often in animals because it is so debilitating, and they don't deal with those mutations as well as plants. We see polyploidy happen a lot more often in plants. Aneuploidy is where 1 or more chromosomes are present in extra copies or deficient in number. So it's a bit like polyploidy, except for instead of an entire multiple set of chromosomes, you have an extra chromosome, or you're missing a chromosome. Sometimes it might be a chromosome or 2, all right? But even just that is more than your body really wants to handle, okay? Sometimes this leads to something very debilitating for the person who's born with it. Other times, nothing really happens at all. Okay, so some examples of these things here of aneuploidy. The most common one, the one that we've heard all about, is Down syndrome. Down syndrome is a result of a non-disjunction event that creates an extra copy of chromosome 21 in all the child's cells. Okay, so every cell in that person's body has an extra copy of chromosome 21. Klinefelter syndrome. We see this same type of thing with non-disjunction events and aneuploidy with sex chromosomes too. Okay, and naturally that leads to like questions on okay, what does that person express, and what do they identify as, and things of that nature. And that's all stuff that I would like to talk about at a later time when we're kind of together in class. So we see non-disjunction events in sex chromosomes as well. There's situations where somebody has XXY, okay? They have all of the genetics of a male and all of the genetics of a female, and in this case, these people are in fact sterile, okay? We have extra Y chromosomes sometimes found in males, okay? These people are called super males. Okay, we'll talk a little bit more about that when we see each other in person. Turner syndrome, people with Turner syndrome, they call this monosomy X. Creates a situation where there's no second X chromosome in the female. This one, a little bit like Klinefelter syndrome, is a lot more debilitating than, let's say, the extra Y chromosome in males. Trisomy X, XXX, they have an extra X chromosome. Again, this one's very similar to somebody who has an extra Y. It almost seems like there's no debilitation from having this extra X chromosome. So depending on where these aneuploidies happen and with which chromosomes they happen to, the effects of it on the individual that have these syndromes can change, can be different. And so you just don't know until you actually see them, see the effect of those things in the person that they happen to be in. Okay. So yeah, Down syndrome, you can see, I just included this picture so you could look at the karyotype, the picture here of all of the chromosomes. And if you look down by the red pointer there, chromosome 21, there's 3 copies of it. Okay, so just that little change in the genetics, even how small chromosome 21 is right here, that little extra one causes all of these sometimes very severe issues in people with Down syndrome. Okay, and you wouldn't think that just one extra copy of one little thing causes these issues, but it really does. And even then, the severity of Down syndrome, it works on like a spectrum, right? There's people who are very high functioning and can live on their own and have a regular job, and then there's people who can't even barely feed themselves, and they have to be in homes to be taken care of. So even then, there's a spectrum of it that is quite interesting. All right, other disorders caused by structurally altered chromosomes. Not going to spend a lot of time on these. I just wanted to throw them out there to let you know that there are examples. One of them here we see called cri du chat. I don't know. I'm not French. I never took French, but that's my guess at it. It's a French term that means cry of the cat. It's kind of sad because the children who get this, when they cry, they sound like cats meowing almost. It's really sad to hear it. Oftentimes these kids are mentally disabled, cognitively impaired. A lot of them die in infancy or early childhood. Very few of them actually make it into adulthood. Certain types of cancers, myelogenous leukemia, CML, is caused by a translocation of chromosomes. Okay, and it causes basically this chromosome here that was normal sized right here has a translocation where it exchanges a high percentage of a large proportion of its chromosome arms with only a small proportion of the chromosome arms from number 9, chromosome 9, and it creates this little truncated thing they call Philadelphia chromosome, and so you can get cancer from that, unfortunately. So those are some of the ways in which your chromosomes can be altered. Oftentimes these wide-scale alterations through chromosome number or through chromosome structural changes can cause some pretty significant issues in the organisms that end up getting them. All right, section 5 deals with other inheritance patterns. Okay, so we're talking about, this entire chapter has been talking about the behavior of chromosomes and chromosome movement and chromosome alterations and all these different things regarding chromosomes. It's also in our best interest here to talk about how some of these there are some other inheritance patterns that are exceptions to this standard chromosome theory that we've been kind of working through all the way from chapter 13 through chapter 14 and now even into 15 a little bit. So there are two normal exceptions to Mendelian genetics that, other than the ones that we've already kind of talked about in chapter 14, but as they relate to chromosome theory of inheritance, one exception involves genes located in the nucleus, and the other exception involves genes located outside the nucleus. Okay? So the first one we'll talk about here is called genomic imprinting, and this is an exception to Mendel's inheritance patterns based on genes located in the nucleus. Okay? For a few mammalian traits, the phenotype depends on which parent passed along the alleles for those traits. You can kind of think of this as almost like sex-linked inheritance on the autosomes. The autosomes are chromosomes 1 through 44, and chromosomes 45 and 46 are the sex chromosomes. So this is almost very similar to sex linkage on the autosomes, meaning that there's some kind of genomic imprint or stamp that is put there by one of the parents. Okay? Genomic imprinting involves the silencing of certain genes that are stamped with an imprint during gamete production. And so it's best outlined here with this little experiment about this mouse here. Okay? This mouse has a trait that allows it to grow into a normal-sized mouse. Okay? And it also has a trait that, if it gets a certain version of it, will cause it to become a dwarf mouse or a small mouse, right? Now, a mouse that is wild type, okay, so this wild type mouse is the normal size mouse. It's the one that's most common in the population. And this normal IGF2 allele will code for a normal size mouse. Okay? Now, if we look at the genetics of this mouse's chromosomes, we see that the paternal one, the one it got from its father, the version it got from its father, is going to matter. Okay? Not yet, but it will matter. As will the version it got from its mom. The fact that it came from its mom and the fact that this version right here, this chromosome, came from its dad, that is going to matter. Okay? When previously we had talked about all examples where it didn't really matter. But in this case, let's just look at the normal Mendelian genetics of the thing. Okay? Mom happened to pass him a normal IGF2 allele, and so did dad. The normal one happens to be dominant. Okay? So he's got two dominant copies of this particular gene, which means he's obviously going to be dominant. He's going to have a normal stature. He's going to be a normal-sized mouse. Okay? Now, let's go to the next one. Let's take this into account. If normal is going to be dominant, okay, and make him normal-sized, then the mutant version of this is going to be recessive, and it's going to cause him to be a small mouse. Okay? So let's look at this example. So normal Mendelian patterns, assuming that. Okay, this one here on the blue chromosome is a normal IGF2 allele, which means it's the dominant version. On the other chromosome, we have a mutant allele, a mutant version of that gene. And in this case, the mutant version would make him a dwarf. But since the normal version is there, it's going to override the mutant version, so he's going to be normal-sized. He's effectively heterozygous for the normal size, being a normal-sized mouse. Okay. Now, you've noticed probably that this diagram has some stuff that I have yet to talk about. It's got the words here expressed and not expressed. Expressed and not expressed. So what that means is that the paternal chromosome, the chromosome that he got from his dad, the version that he got from his dad, is always going to be expressed, regardless of what's in mom's version. Okay? And so that manifests now as we look at the final example. The final example is also heterozygous. Okay? This middle example was heterozygous, and we expected him to be normal-sized, right? This one is also heterozygous. We notice that mom gave him a normal version and dad gave him a mutant version. Okay? So under normal Mendelian genetics, we would expect mom's normal version to override the mutant version given by dad. But what we see is totally contrary to that. What we see is a dwarf mouse, a mutant mouse that expresses the recessive phenotype. And the reason for that is because this mouse's father gave him the mutant allele. And that's the only one that gets expressed. Okay? This version given to him by his dad is the only one that will ever be expressed. So if we go up here to these other two examples, it wasn't the fact that this normal version was overriding the mutant allele. It was the fact that mom's mutant allele never got expressed in the first place, and the only one left to be expressed was dad's. So that's what we see manifest down here. Even though mom gave him a normal version, and under normal Mendelian genetics, that one should override the mutant allele, just doesn't. It's not allowed to express itself. Because in this case, dad's is expressed. And dad's will always only be expressed in this example. This is what we call genomic imprinting. Okay? Effectively, one of the genes is silenced, and the other one is the only other one that's allowed to be expressed. Okay? I hope that made sense. If it didn't, please write down any questions that you had about it. Additionally, and this is an example of extracellular genes or extracellular inheritance, we see, and this is not happening in the nucleus, right? These are genes that are found outside of the nucleus. These are genes found in organelles in the cytoplasm. And there's effectively two places that we see genes that are found outside of the nucleus. We see them in the mitochondria, and we see them in chloroplasts. Both of those organelles actually have their own DNA. Okay? So the inheritance of traits controlled by extracellular genes depends on the maternal parent, because the zygote cytoplasm comes from the egg. Okay? And I'll throw up some diagrams to kind of show you what I'm talking about here. Okay? So the genes in the mitochondria are passed on from mother to child. Okay? All of the genes that you see in you that are found in the mitochondria in you come from your mother. And in fact, this is a way, regardless of what you believe in terms of Bible or religion or whatever, this is theoretically an argument for an actual Eve. Okay? So think about it. If all of the mitochondrial DNA that I got is from my mom, where do you think she got all of her mitochondrial DNA? From her mom. And where did her mom get all of her mitochondrial DNA? From her mom. And so on and so forth. So in theory, and there's been documents done about this, with documentaries done about this, I think through National Geographic, they have been able to look at mitochondrial DNA and trace it back to the very first human female. Okay? It's actually quite fascinating. Now, these mitochondrial DNAs, they don't do a ton, but they code for genes that create polypeptides or proteins that are subunits of enzyme complexes of the oxidative phosphorylation system. So what the heck does that mean in English? That means that the mitochondrial DNA, the DNA that's found in the mitochondria, makes the little proteins that are found in the ETC, the electron transport chain. They make the little proteins that make the Krebs cycle run, okay? Because all that stuff happens in the mitochondria. So effectively, you could say that I've got my mother's metabolism, okay? And my mom is, she's quite a small lady, and she's stayed that way. And I have kind of stayed that way myself. I tend to burn through food really quickly, so it kind of makes sense. So that's kind of a neat way to look at it. Now, this diagram, I think, helps us understand a little bit more of the inheritance of mitochondrial DNA and why we only get mitochondria from our parents, or from our mother. The picture we see on this side of the pedigree, this is effectively a pedigree, shows what the oocyte is the egg, looks like. The oocyte has all of the nuclear DNA that's going to be passed to the child. It also has all of the cytoplasm and the organelles and the mitochondria that the kid is going to need. Because look what Dad supplies. Dad supplies almost nothing. Okay? The size of the sperm from the male is far, far smaller than the actual size of the egg given by the female. Okay? And if you look at what's going on here with the male, there's really nothing else inside the sperm other than DNA. There's a little mitochondria here so that this thing actually has some energy to make the tail run, but that's it. Okay, it comes with no cytoplasm, it comes with no organelles, it comes with a little thing to use energy and a little thing that packages up DNA. When it fertilizes the egg, mom supplies everything else. So every child, regardless of whether or not they're male or female, is going to have the mitochondria that originally came from mom. So that is how the inheritance of mitochondrial DNA works and why we only get that mitochondrial DNA from our mothers. Okay? So that does it for chapter 15. This last one is a little bit more lengthy, but I think it was best to get it all done in one fell swoop. If you have any questions, obviously feel free to write them down, go back and review, write them down, and then please ask on anything that you have. And I will see you guys shortly.